Hello friends, welcome back to our YouTube channel Doctor's Corner. Today we are going to discuss about hemostasis. It is a very important topic as far as theory exam in physiology is concerned. Many a times a long question has been asked in the theory university exams regarding hemostasis. Okay, so today we'll study about this hemostasis. First, what is hemostasis? Hemostasis, as the name indicates, hemo means blood, stasis means simply stoppage, that is prevention of bleeding. Okay, so whenever there is a cut or injury to a blood vessel and there is a bleeding, that is blood is oozing out, then some regulatory mechanisms in our body will help to prevent the bleeding, and that mechanism is known as hemostasis. Okay, so there are various steps of hemostasis we are going to discuss in detail regarding this. So the first step is in the hemostasis is vasoconstriction. As soon as the bleeding starts, okay, vasoconstriction, okay, there is the vasoconstriction. Then the second step will be formation of the platelet plug. Okay, so out of the three important cells in the blood, that is the RBCs, platelets and WBCs. So platelet is very important, formation of the platelet plug. Then finally the blood clot formation, that is the blood coagulation. This process is known as blood coagulation. So only the blood coagulation can be an important question or a short note in physiology theory examination. Then finally there is a clot retraction or fibrous tissue formation. Okay, so we'll study this process of hemostasis, that is the stoppage of the bleeding from a injured blood vessel under this side headings. Okay, that are the steps. So now to understand this, let us describe this with a diagram. Now I am just drawing roughly a blood vessel. Okay, consider this as a blood vessel, and you know the lining, the cells lining, it is known as endothelium. Okay, so this is endothelium of the blood vessel. So endothelial cells which are lining the a blood vessel is just a normal blood vessel, and inside this there is a blood plasma and various cells. The important cell here is the platelets. Okay, so this hemostasis is concerned with the platelet, the important cells, not what we are going to discuss today about the platelets, or the indirectly we are discussing the functions of the platelets. So these platelets are small cells around 1 to 4 micrometer in size or diameter. Okay, and the normal platelet count is around 1.5 lakhs cells to around 3 lakhs, more specific 3 lakhs cells per cubic millimeter of the blood or per microliter of the blood. Okay, so platelets is an anucleated cell, that is there is no nucleus present in the platelets as similar to RBCs. Okay, and but it has some additional things, like it has a contractile protein same like skeletal muscles, actin and myosin, and also thrombosthenin. Okay, apart from that it has a well developed endoplasmic reticulum, Golgi apparatus, mitochondria. Then in the platelets there are certain granules. Okay, we discuss about this granules, alpha granules, delta granules and all, and this platelet membrane, okay, the plasma membrane of the platelets, okay, that has a phospholipid and it also releases so many chemicals and all, we'll discuss today. Okay, so this is all about platelets and the hemostasis. Okay, now consider there is injury to this blood vessel. Okay, there is injury or damage to this blood vessel and blood is oozing out. Okay, blood is oozing out. So it can be a pin, a needle stick or anything, any prick to the blood vessel, and there is an injury and the blood is oozing out. Okay, so where it will come? It will come to the extravascular tissue. So mainly the extravascular tissue, it has the collagens, and again there are the smooth muscles and all will be present, right? So now whenever there is an injury, what are the process that takes place? Okay, so in the hemostasis, so first step, as I already said or described, that is the vasoconstriction. So first thing what will happen is vasoconstriction. Now what is vasoconstriction? So simply there is a constriction of the blood vessel. So constriction means the diameter of this blood vessels will decrease. There is a vasoconstriction. Now why this vasoconstriction will occur? What are the factors that are responsible for this? Is it can be due to neurogenic reflex or nervous reflex. Okay, can be nervous reflex. Okay, so there are nerve endings here. Okay, there are nerve endings here and all, and reflexly, nervous reflex, there is a vasoconstriction, or there are smooth muscles present outside these blood vessels, so it can be a myogenic spasm, or finally some chemical substances, some autocoids, okay, like endothelin, okay, some chemicals and all, autocoids or endothelins can cause us the vasoconstriction, or substances which are released from the platelets like thromboxane A2. Okay, thromboxane A2. This is also a vasoconstrictor substance. So this all factors will cause us the vasoconstrictions. Now this endothelin is actually released from this endothelial cells. Okay, first step is vasoconstriction. Okay, so that the blood which is oozing out, okay, so to some degree blood vessels as a reflex mechanism try to prevent the blood loss. First thing. Second step is platelet plug formation. Here the role of the platelet comes. So platelets, what they will do is they will form a plug here. Okay, so these platelets which are circulating here normally in the blood, they will come and they will bind and they will form a plug like this. So this process is known as platelet plug formation and try to seal the damaged blood vessel. Now how they will do it? Again, there are steps here. How the platelets bind here and they will prevent the bleeding. Let us discuss one by one what are the steps here for platelet plug formation to the damaged blood vessel. Okay. Now first one will be platelet adhesion. Platelet adhesions. Okay. So normally the platelets which are circulating in the blood vessels, they will not come and stick to the endothelial surface. Okay, there are certain anticoagulant mechanism which we'll discuss later. Okay, they will repel and they will not stick to this endothelial cells. But whenever there is a damaged endothelial cells or the ruptured endothelial cells, there are certain proteins which are released here, from the damaged endothelial cells and from the plasma. One such protein that will come here is, this is known as von Willebrand factor, okay, which will come from this damaged endothelial cells. Now these platelets will come and adhere or stick to this von Willebrand factor, okay, that is known as platelet adhesion. Okay, so let me describe here. So consider this blue color molecules are the von Willebrand factor proteins, okay, which are released from the platelets, and also they are present in the plasma, which leaks out to the site of the damage, and this black color cells are the platelets. Okay, now on the surface of the platelets, that is on the plasma membrane of this platelets on the membrane, so there are certain receptors, glycoproteins mainly, so they are known as glycoprotein 1b. okay, these glycoproteins normally does not binds to the healthy endothelium, but in the damaged endothelium where there is a von Willebrand factor, they go and stick here. Okay, and they go and stick, and this process of sticking of the platelets to this von Willebrand factor and or the collagen, this process is known as simply platelet adhesion. Okay, where it will come and stick here. Okay, now once they come and stick to this, there is a second process in the platelet plug formation that is known as platelet activation. So platelets will get activated once they are bound here, so the platelets will get activated. So inside these platelets, I said you there are granules, okay, some alpha granules and some delta granules. So the delta granules will secrete certain substances like serotonin, ADP, okay, and calcium. Okay, alpha granules will secrete platelet derived growth factors and some clotting factors and all. Okay, we'll discuss later about these granules, but these are some important granules. Also, from the surface of the platelets, from the phospholipids and all, ultimately the arachidonic acids are converted in by the cyclooxygenase enzyme into very important substance known as thromboxane A2. Thromboxane A2 and ADP, these are two important substances which will help in the platelet aggregation, that is the next step. So platelet activation, once they are binded after the adherence, they get activated, and activation means they will release of so many chemical substances, mainly ADP and thromboxane A2. This thromboxane A2 has two roles. It can act as a platelet aggregator and helps in platelet aggregation and also it can act as a vasoconstrictor. So platelet aggregation is the third step in the platelet plug formation. So what happens during this is, so the platelets will try to stick to each other. So again, on the surface of the platelet there is one more protein, glycoprotein GP2B3A. Okay, so this glycoprotein 2B3A will bind the platelets or stick the platelets to each other. That is, the process of sticking of the platelets to each other is known as aggregation, and process of sticking of the platelets to the damaged endothelium on the van Willebrand factor, it is called as platelet adhesion. Adhesion is different, aggregation is different. Okay, now once it is activated, there is platelet aggregation. So platelets, normal platelets, surrounding platelets, they will aggregate here. Okay, they will aggregate here, thromboxane A2 and ADP. Okay, these chemical substances will help you. Even thromboxane A2 will cause vasoconstriction. So once they are aggregated, they form a platelet plug. That is the second process, that is they form a platelet plug. So for platelet plug, what are the steps? Platelet adhesion, then platelet activation, and platelet aggregation. So once this platelet plug is formed, it will try to seal the ruptured blood vessel. Okay, but it is a very it is a weak structure, not a very strong structure. We need to make it strong. Okay, that is the next process, that is the blood coagulation or the clot formation. This is just it is also known as temporary hemostatic plug or the primary platelet plug or primary hemostatic plug. So coming back to the starting side headings, we have discussed about vasoconstriction, platelet plug formation, then third step is blood coagulation. Now blood coagulation itself can be a 10 marks question for you in the theory exams, very important topic. So blood coagulation means, this what we have discussed till now is the process of hemostasis, that is the process in the stopping of the blood. Now actual clot formation. So actual clot formation means formation of this meshwork of fibrin, network of fibrin mesh. This process of formation of the fibrin mesh from a plasma protein fibrinogen, this process is simply known as blood coagulation. Okay, now how exactly this fibrinogen is a substance, a plasma proteins, is converted to this fibrin mesh, okay, and form the blood coagulation or blood clot. So what exactly happens is, so in this platelet plug, temporary hemostatic plug or this platelet plug, which is a weak one, so this fibrin mesh forms and it will entrap these platelets and the other cells like RBCs, WBCs and the plasma and the clotting factors, they will get entrapped, and this mass is known as blood clot or the blood coagulation. Okay, now how this fibrin threads or the fibrin mesh is formed? Okay, it is formed by some clotting factors which are present in this plasma or the liquid part. So in this plasma, the property of the blood coagulation is by the plasma. So plasma has certain plasma proteins like fibrinogen, okay, and prothrombin and all, so many things are there. So that will ultimately form this. So alone plasma is capable of forming the blood clot, okay, along with the clotting factors. So there are so many clotting factors, some chemicals are present here. So they are named according to the Roman numbers. Okay, up to factor 13 is there. Okay, and other clotting factors are there. For example, factor 1 is fibrinogen, which is a plasma protein. Okay, that is only is converted into fibrin thread, that is known as factor 1. Factor 2, Roman number factor 2 is prothrombin. Factor 3 is tissue factor, tissue thromboplastin. Factor 4, calcium. Okay, similarly we have so many things. The important ones are factor 8, anti-hemophilic factor, deficiency of this causes hemophilia, one disease. Okay, factor 9, Christmas factor. Okay, then factor 12, Hegman factor. Factor 13, fibrin stabilizing factor. So many things are there, along with high molecular weight, kininogen, prekallikrein. Okay, and platelet itself is a clotting factor. So these are the 13 clotting factors plus other things. Okay, now these chemicals are normally present in the blood. Now these chemicals form a cascade of chemical reactions to form this fibrin thread. Now we have to discuss that cascade of chemical reactions, how this will lead to ultimate formation of this fibrin mesh or fibrin thread. Normally in circulation there will be fibrinogen in the plasma, but this is converted once there is a platelet activation and platelet plug formation. Now fibrin is converted into fibrin. Again, in this blood clot formation there are 3 steps. First is formation of prothrombin activator. First step is formation of prothrombin activator. Formation of prothrombin activator. Second is conversion of prothrombin to thrombin. Okay, so a prothrombin activator, one chemical substance or chemical complex is formed that will catalyze the conversion of prothrombin to thrombin, and finally this thrombin will convert fibrinogen into fibrin fibers or fibrin mesh. Okay, that is the blood clot. Fibrinogen is converted to fibrin mesh, that helps in formation of the blood clot. So there are three steps. This fibrin mesh or fibrin strands are not formed directly, it is formed by the so many chemical reactions. We'll see one by one. Now this is the most rate limiting step or the lengthiest step, formation of the prothrombin activator. So some chemical reactions takes place. It takes place by two mechanisms. One is known as intrinsic mechanism, another one is known as extrinsic mechanism. Intrinsic mechanism is activated whenever there is a trauma to the blood itself, or whenever some clotting substances gets closely attached to the damaged endothelium. When they come in contact with the damaged endothelium, they get activated. Whereas extrinsic factor means whenever there is a trauma to the blood vessel or the extravascular tissue, some extrinsic factors will cause the activation of certain clotting factors. This all takes place in the blood itself. Okay, this cascade of events. Okay, so first, intrinsic mechanism, whenever there is a blood trauma or when the clotting factors are exposed to the damaged endothelium, or even when you draw the blood in a clean test tube and all, where there is a blood clot, it is intrinsic mechanism. That is, some intrinsic factors causes the blood coagulation. Okay, when they are exposed to the electronegatively charged glass substances. Okay, so here factor 12 is the most important one, which was in inactive form normally in the circulation in the blood, but when all this process, platelet activation and all starts, it gets activated. So we write a symbol small a here. Factor 12 gets activated as factor 12 activated. Okay, now this factor 12, what it will do is it will cause the activation of factor 11th. Okay, it will causes the activation of the factor 11th. Okay, now this factor 11th will causes the activation of factor 9th. Okay, factor 9th A. So first I will try to explain this, then I will show this diagram also. Okay, I'll try to show this diagrammatic representation how this will happen, but before that I'll just try to explain these things. Okay, so factor 12th gets activated as activated factor 12. Okay, then this will catalyze the inactive factor 11th to active factor 11th. For this, high molecular weight kininogen, okay, or prekallikrein is also used, okay, for this particular reaction. Then factor 11th converts the factor 9th. Okay, 10th is not there, 10th will come later. Then this activated factor 9th will causes the conversion of inactive factor 10. That is the most important step. Okay, common pathway. Factor 10th inactive gets converted into active factor A by the factor 9th, but along with this some substances are required, that is factor 8th. Okay, calcium molecule. Okay, calcium, here also calcium was important, calcium. Then some phospholipids, platelet phospholipids. So all these substances are required for the conversion of the inactive factor 10 to factor 10, activated factor 10. Okay, this is very much important. Now finally this factor 10th, okay, factor 10th, this factor 10 along with factor 5th, okay, factor 5th, calcium and phospholipids, platelets phospholipids and the tissue phospholipids, this form a complex. And that complex is simply called as prothrombin activator. Prothrombin activator. What is prothrombin activator? This activated substances combination, that is activated factor 10th, factor 5th, calcium and phospholipid, together they form an complex that is known as prothrombin activator complex. So our first step in the blood coagulation is what? Formation of prothrombin activator. What is prothrombin activator? Activated factor 10, factor 5th, calcium and phospholipid. Okay, now prothrombin activator convert the prothrombin to thrombin, and thrombin will finally will also catalyze this, and it will activate the factor 5th. Initially factor 5th was not active, but when thrombin forms, it will activate the factor 5th also, and this process will continue. This positive feedback mechanism. Now what we have discussed is the intrinsic mechanism. There is something known as extrinsic mechanisms also. Extrinsic mechanism, whenever there is a damage to the blood vessel or trauma to the extravascular tissue, so what happens is tissues will release certain thing known as tissue factor, which is the factor 3, that is tissue thromboplastin. This tissue factor or the tissue thromboplastin will convert the factor 7th into activated factor 7th. This factor 7th activated factor 7th ultimately convert the factor 10th into activated factor 8. Okay, and the rest of the story is same like this, that is it will form the prothrombin activator. Let me show you this diagrammatic representation here. So tissue factor will convert the 7th to activated 7th, and this activated 7th either it will activate the 9th or directly the 10th. Okay, and this 10th along with the factor 5th, calcium and all, they form the prothrombin activator. Okay, now by extrinsic or intrinsic pathway, ultimately prothrombin activator is formed. Okay, till 10th it is a common, after 10th it is the common pathway. Till 10th a intrinsic and extrinsic is different. You can see the extrinsic pathway is bit small, only the factor 7th and directly and tissue factor 3s are involved. Okay, factor 3 are involved till activation of factor 10th. Okay, whereas for intrinsic pathway such a big cascade is there. Then the second step here in the blood clotting factor is conversion of prothrombin to thrombin. So prothrombin is a plasma proteins having a molecular weight of 68,700, gets converted into thrombin. Okay, thrombin is a low molecular weight substance, almost half of this, around 33,700 molecular weight. Okay, so conversion of prothrombin to thrombin requires prothrombin activator. So this prothrombin activator converts prothrombin to thrombin, and finally this thrombin acts as a protease and converts the fibrinogen, a plasma protein, into fibrin monomers. So how exactly it might be, I'll show you. So fibrinogen is a protein, okay, plasma proteins. So they gets disintegrated by this protease thrombin molecule will break this fibrin molecule, okay, which was a soluble molecule, into fibrin monomer, small, and they has a tendency to polymerize. They form a chain like this. Polymerization means they will unite and form a chain. This is known as fibrin polymerization, and ultimately with the help of the factor 13, that is fibrin stabilizing factor, this fibrin forms a mesh network that is known as fibrin fibers. So ultimately fibrin fibers are formed by the fibrin stabilizing factor and calcium and phospholipid, and fibrin fibers are formed. These fibrin fibers along with the entrapped cells forms the blood clot. Okay, hope you understood this. So ultimately blood clot is formed by these fibrin fibers and the entrapped cells, that is RBCs, WBCs, platelets and the clotting factors. So after that there is known as clot retraction. So it will try to squeeze. After 30 minutes the contractile proteins like actin, myosin and thrombostatin, which is present in the platelets, it will try to contract. Now this will contract and there is a squeezing of some fluid that is known as serum. Okay, so what is serum? Serum is plasma minus clotting factors. Serum is plasma minus clotting factors or fibrinogen clotting factors. So clotting factors are utilized in the formation of the blood clot. So when they contract, the fluid which is released is serum. Okay, so this is all about the blood clotting. Let me summarize what we have discussed today. Hemostasis, that is how the process of stoppage of the bleeding from a ruptured blood vessel takes place. That is, hemo means blood, stasis means stoppage. So this process takes place by vasoconstriction initially, then platelet plug formation, blood clot, and clot retraction finally. So vasoconstriction we have this did, then platelet plug formation, it requires platelet adhesion, platelet activation, then platelet aggregation. Then third step is the blood coagulation. We have this 13 Roman numerical clotting factors along with prekallikrein, high molecular weight kininogen, and platelets. Okay, then intrinsic and extrinsic pathway. Then after that, ultimately by the intrinsic and extrinsic pathway, prothrombin activator is formed which will convert prothrombin to thrombin. Then thrombin will convert fibrinogen to fibrin monomer. Fibrin monomer in presence of calcium form the fibrin fibers, and in presence of factor 13, that is fibrin stabilizing factor, it forms cross-linked fibrin fibers which is blood clot. Okay, then finally there is clot retraction. Okay, we will discuss about the other applied aspect, that is antiplatelet drugs and anticoagulants in some other videos. Hope you like our lecture. If so, please subscribe our channel and share the link with your friends. Thank you.